Everybody. We're kind of here in the home stretch, so I thank you all who are still hanging in there. I know that this promises to be a very stimulating discussion and presentations. So the name of this session is Cancer, Latest Breakthroughs and Discoveries, but we're continuing on the momentum of an earlier session on Immuno-Oncology. So the focus will be along the lines of cancer immunotherapy, including stem cells. And so our first speaker is Dr. Malvina Prapa. She is a fellow at the Laboratory of Cellular Therapies, University of Modena in Italy. And the title of her talk is Adoptive Chimeric Antigen Receptor, CARs, as you all know them, T-cell therapy to target GD2 positive cancers. So please welcome Dr. Prapa.
Okay. Thank you for the introduction. It's a pleasure to be here, and I'm glad to share with you some research data. The topic of my presentation focuses on CAR T-cell therapy to treat cancer, specifically GD2 positive cancers. The rationale of this approach, once it gets translated into a clinical setting, is to use T-cells previously isolated from the peripheral blood of the patient, ex vivo gene modified with a chimeric antigen receptor, then expanded in order to be reinfused back into the patient. This is a short introduction on the modular structure of the chimeric antigen receptor. It is a modular structure, as I say. Here is represented the first generation structure, which consists into a single chain extracellular domain of binding to the antigen of the interest, which is derived from the variable fragments of the heavy and light chain of a monoclonal antibody specific to that antigen. The transmembrane domain and the intracellular domain of signaling, which is derived from the CD3 zeta chain of the T-cell receptor. This structure has then evolved into a second and third generation, which novelty consists into the introduction at the intracellular domain of a costimulatory molecule, as represented in here, and of two combined costimulatory molecules in here. So once the transduced T cells with chimeric antigen receptor has been obtained, they are supposed to be able to recognize and to bind the tumor-associated antigen of interest in an MHC-independent manner. This is one of the major turning points of this technology, since we know that tumor cells often downregulate or lack the expression of such complexes, escaping this way the immunorecognition and giving rise to clinically over tumors. So once the binding takes place, the chimeric antigen receptor will mediate the signaling into the T cell, which in turn will activate all the cytotoxic mechanisms in order to mediate the killing of the tumor cells. Of note, chimeric antigen receptors can potentially be redirected versus a wide range of biological structures, such as protein, lipids, carbohydrates. In this view, we have focused our attention on this diallogagliocyte G2 as therapeutic target for CAR T-cell purposes, since it is low expressed in normal tissues but retain high levels among pediatric and adult solid tumors, as represented in here. So in our study, we have focused our attention on neuroblastoma and recently on glioblastoma tumors. They are really aggressive tumors. Neuroblastoma is the most common extracranial solid tumor in childhood. At diagnosis, almost 50% of the patients represent a metastatic disease. For this high-risk patient, the 5 years survival rate is less than 30%. So in this perspective is of pivotal importance exploring new strategies, new therapeutic strategies, in order to keep under control the minimal residual disease in this patient following conventional therapies. Among a few anti-GD2 CAR have been explored, we have proposed a second-generation CAR here represented in this cartoon, which novelty consists into the extracellular domain, which was derived from an IgM class, from the single-chain variable fragments of an IgM class anti-GD2 monoclonal antibody. In stream, it contains a potent costimulatory molecule in the intracellular domain, such as 4-1BB. This structure has been then introduced by retroviral vector in T cells. Here I show you GD2 CAR T cells derived from two different donors. In here was assessed the transduction efficiency, both detected by both GFP expression as well as the chimeric antigen receptor. This is our T cell, our control T cell population. Then the immunophenotype was assessed, as I show you in here. There is a significant increase in two cytotoxic subpopulation in both the transduced cells in respect to the non-transduced parental T cells, such as CD8 positive cells, as well as cytokine-induced killer cells, prompting their use into functionality studies over several cytotoxic assays. This is a standard chromium release assay. The rationale is the higher radioactivity readout you get, the highest cytotoxic effect you have mediated by the GD2 CAR T cells in co-culture with a GD2 positive cell line in respect to the GFP-only T cell, which is our control. For the GD2 negative cell line and for control HeLa cells, there was no difference between the GD2 CAR-treated cells in comparison to the control condition. This is the viability assay. The rationale is that the lower fluorescence readout you have, the lowest viability, or better, the lower, the less live tumor cells still persist after 4 hours of co-culture with GD2 CAR T cells in respect to the controls. Overall, the effector to target ratio we assessed. For the GD2 negative cell line, there was no difference. We were able to detect over all the condition, the co-culture condition. In here, we assessed a 48-hour co-culture with an enriched population of GD2 CAR T cells, which we achieved by FACS sorting, in co-culture to effector to target ratio of 1:1 with the GD2 positive cell line. As you can appreciate in this graphic, there is a huge cytotoxic effect mediated by GD2 CAR T cells in respect to the control one. This activity was then associated with a potent release of two pro-apoptotic molecules, such represented in this graphic in respect to the control. So then we moved into the in vivo study, a neuroblastoma model with not-skate mice. Animals were given 1 million of GD2 positive cell line, as well as the first dose of either GFP-only T cells or GD2 CAR T cells in effector to target ratio of 5:1. The second and third dose was given respectively at day 7 and day 14. Animals then were monitored till day 28. In here, I show you a representative tumor burden occurred per each group. This was the unique tumor burden we were able to collect in the GD2 CAR T cell treated group. On contrary, for the other two groups, in all animals, there was tumor generation. This is the histologic study in which we wanted to detect persisting and infiltrating cells into the tumor, detected by GFP signal. As you can see in here, there are infiltrating niche in both T cell treated group, but exclusively GD2 CAR T cells were able to almost abrogate the tumor growth. As you can see by the difference in the tissue architecture in respect to these two specimens. Then we assess the apoptosis rate by tunnel assay. As you can see here, there is as well difference between this specimen in comparison to the other two. Here we detected the GFP signal in blood samples, which were collected at sacrifice date, suggesting that circulating T cells still persist at least at day 28 after the treatment. So collectively, our preclinical data are suggesting that anti-GD2 CAR is able to mediate a specific cytotoxic activity versus GD2 positive neuroblastoma cells. This activity is associated with a potent release of pro-apoptotic molecules. In vivo, T cells were able to infiltrate and persist into the tumor mass, as well as to persist into the blood circulation until day 28, but exclusively GD2 CAR T cells were able to abrogate the tumor growth. Recently, we have focused our attention on glioblastoma tumor model. It is a really aggressive tumor, is a high-grade astrocytoma. The prognosis for the patient is really poor, as usually the survival rate doesn't exceed 1.5 years. So again, here we were able to detect a specific targeting versus GD2 antigen in the GD2 positive cell line in co-culture with GD2 CAR T cells in respect to the control over all the condition ratio after 4 hours of co-culture, as well as after 8 hours of co-cultures. For the GD2 negative cell line, there was no difference between the treated and the control condition. Here I show you a 3D model, a 3D culture of GD2 positive glioblastoma spheroids in culture alone or in co-culture with either GFP-only T cells or GD2 CAR T cells in effector to target ratio of 1:1 after 72 hours of co-culture. As you can see here, we were able to appreciate the difference in the spheroid dimension when treated with GD2 CAR T cells in respect to the GFP-only T cells and in comparison to the spheroids with no treatment, suggesting again that there is a specific targeting. Now we have a collaboration with a laboratory of brain tumors here at Upstate Medical University, where I'm gaining some training in the intracranial surgery in order to develop in vivo GD2 positive glioblastoma models, then after to challenging them with this approach. To conclude, our goal is to translate this approach into a clinical setting in order to target the minimal residual disease in high-risk patients affected by GD2 positive cancers. I would like to thank my laboratory, our collaborators, my host laboratory here in the United States, and thanks so much for your attention.
Thank you, Malvina. Clearly, your results show the promise of CAR T therapy for solid tumors, especially the very difficult brain tumors. Very, very interesting. So moving on to our next speaker, Dr. Shannon Murray. Dr. Shannon Murray is an assistant professor at the Cell Therapy Institute at Nova Southeastern University in Fort Lauderdale. And the title of her talk is Overcoming the Immunosuppressive Tumor Microenvironment. So please join me in welcoming Dr. Murray to the podium.
Okay, and thank you so much to the organizers for inviting me to speak today. And I'm going to be talking a bit about advances in overcoming tumor microenvironments that are immune suppressive, as well as talking about some recent research in my lab in collaboration with Andreas Lundquist at Karolinska Institute. So I think all of you know that now we don't really think of a tumor as a homogeneous mass. We understand that it's actually a heterogeneous mix of tumor cells in addition to immune infiltrates. And we know that these immune infiltrates, when we look really more closely, they have these phenotypes of being immunosuppressive, namely T regulatory cells, myeloid-derived suppressor cells, which inhibit the differentiation of myeloid cells into dendritic cells, as well as the accumulation of immunosuppressed natural killer cells in this environment. So some recent advances have really focused on the use of combination therapies in combination with the checkpoint blockade inhibitors, such as the anti-PD-1 and anti-CTLA-4 immunotherapies, in combination with components that will overcome the immunosuppression that occurs in tumor microenvironments. I won't talk a lot about it today, but some of the new advances are also understanding how the microbiome actually plays a role in the efficacy of these immunotherapies. It appears that some of these checkpoint blockade inhibitors actually interfere with dendritic cells in the gut, sort of breaking some of the tolerance mechanisms. So again, as I said, tumor cells themselves, cancer cells within the tumor, secrete immunosuppressive factors that in turn suppress other immune cells. And again, back to the checkpoint blockade inhibitors, the way that these work is the tumors often are upregulating PD-L1 and then interact with the PD-1 on the T cell, resulting in an exhausted T cell with a phenotype of being unable to produce granules and lyse target cells. So a lot of the tumor cells themselves secrete these immunosuppressive factors, induce these myeloid-derived suppressor cells, which in turn produce a lot of factors that are immunoinhibitory. There are also T regulatory cells that are induced in this environment. So we have not only the tumor cell inducing, secreting immunosuppressive factors and upregulating receptors, but then we also have these immune cells now that are impaired, including natural killer cells. So I want to talk about a few areas of research in the lab, and then just a few updates. So basically, I'm going to actually turn, I can see better over here, I'm sorry. Basically that here we have, well, but all of you are over here. Okay, I'm going to go over here. I can't see really wonderfully, but I'm going to talk from over here. So some advances in T regulatory cells are very interesting. Basically, some groups, particularly Shimon Sakaguchi and has basically really identified some subsets of T regulatory cells that seem to be the pathogenic subsets, and that they express CCR4, so development of some antibodies that can be used to deplete Tregs in the tumor microenvironments are being pursued. I'm going to talk quickly about a drug therapeutic, a therapy that we're looking at that decreases T regulatory cells in the tumor microenvironment. I want to talk a little bit briefly about myeloid-derived suppressor cells in the centrality of prostaglandin E2 secreted by multiple tumor types to induce the cell type, which in turn suppresses T and NK cells. So basically, I want to talk a little bit about some of the progresses using prostaglandin E2 or cyclooxygenase inhibitors in combination with these checkpoint blockade inhibitors, which is an area of further research, as well as some work of developing an in vitro model to study myeloid cell differentiation and effects with tumor-derived factors. But I want to start with some of our work in collaboration with Dr. Andreas Lundquist, who's a visiting professor at Nova Southeastern at the Cell Therapy Institute, as well as at Karolinska Institute, and talk about some published work first about using IL-15 to improve natural killer cell anti-tumor responses. And a few other areas of ongoing research are how to engineer natural killer cells to overcome the hypoxic tumor microenvironment, as well as get them to the correct sites after being used for adaptive therapy. So first, in some published work regarding IL-15 priming of natural killer cells. Now, I think you've heard a bit in the sessions earlier about natural killer cells, but this model is making use of an autologous natural killer cells for solid tumors. And basically, there's been a lot of evidence that the ability of the natural killer cells to persist play a role in cancer clearance. So these studies were evaluating IL-15, versus IL-2, which has been the standard way to expand autologous natural killer cells in vitro. But then what was added to this study was actually looking what occurs after cytokine withdrawal of the NK cells post-expansion. So when you look during this initial priming, you see a very similar ability of these natural killer cells to kill, whether they were expanded in IL-2 or IL-15. But when you look at the cytokine withdrawal period of 48 hours, you see a differential effect, both in the ability of the NK cells to lyse tumor targets, as well as the natural killer cells to be resistant to apoptosis. So we did some genomic studies using translitome studies where we looked at total cellular RNA as well as the polysome-associated RNA. And what we found was in the prime, during those NK cells that were primed with IL-15 versus IL-2, had a profile of upregulation of mitochondrial-associated genes and cell cycle-associated genes. And these mitochondrial genes and metabolism genes we wanted to look further into. We did some studies where we looked at glycolysis rates between the IL-15 stimulated NK cells versus the IL-2 stimulated, and saw an increased glycolysis in the IL-15 primed NK cells. And additionally, unlike the IL-2 primed NK cells, this was dependent on the mTOR pathway by using taurin as an inhibitor. Finally, in summary, that work really addressed whether IL-15 might be one way to improve natural killer cell function in tumor models. Finally, in an NSG immunosuppressed mouse model, more of the IL-15 primed NK cells home to the liver as opposed to IL-2 primed NK cells. So in summary, these metabolic processes after cytokine withdrawal were improved by IL-15 as compared to IL-2. So I wanted to share those because of the importance of adoptive transfer of NK cells into tumor immunotherapy. Okay, so I think we've heard a lot about this. Not all tumors are equal. There are distinct kind of immunologic control between different kinds of tumors, and I'm just highlighting renal cell carcinoma here and melanoma. And just to highlight, for example, when we can look at studies and prognoses, we can see that CD8 T cells infiltration into the solid tumor is associated with improved prognosis and is actually associated with poor prognosis in the case of renal cell, whereas natural killer cells are much more associated and there's a strong association with improvement of renal cell carcinoma. So additionally, the myeloid-derived suppressor cell compartment is of interest. And what we see is, so there are two major dominant types, and more are being delineated, of myeloid-derived suppressor cells, both of granulocytic origin and of monocytic origin. And we see a greater accumulation and association with prognosis of the different types in the different cancers. For example, the monocytic MDSCs are associated with poor prognosis in melanoma, and we see an accumulation in renal cell carcinoma of the granulocytic. In both cases, we see T regulatory cells are associated with poor prognosis. However, these cell types, I'm focusing on the tumor microenvironment with myeloid-derived suppressor cells. They also accumulate in the blood, and the blood are a good biomarker for tumor microenvironments. However, we don't always see the same thing with Tregs. Tregs within the tumor and with the blood did not always correlate in cancers with the prognoses. So I just want to briefly talk again. I know this is the World Stem Cell Summit, so I wanted to talk a little bit about cell differentiation and ways that we can differentiate cells to become a cell type that's a more immune potent in a tumor. So this is a schematic of myelopoiesis, starting with the hematopoietic progenitor, going to the granulocytic arm, as well as the monocytic and DC arm, and just show you here tumor cells secreting things that lead to these directions in red arrows. So basically what happens during many tumors is this preferential differentiation into the granulocytic arm, and also away from dendritic cells and potent macrophage and to these myeloid-derived suppressor cells, as well as of these other immunosuppressed suppressive myeloid cells. So basically, again, these myeloid-derived suppressor cells, they potently inhibit T and NK cells, and blood is a good biomarker for these. So you can see that the presence of the tumor and factors that are directly secreted by the tumor skews myelopoiesis away from some of these more potent cells, such as dendritic cells. So we've developed an in vitro model, and this was a model developed by Yuming Mao in Dr. Rolf Kiesling and Andreas Lundquist's lab to study basically the interaction between myeloid cells and tumors, and making use of an early passage melanoma cell line. Mao was able to really see from CD14 positive monocytes from healthy donors, see a shift to MDSCs and away from dendritic cell development, and able to see suppression by these MDSCs of NK and T cells. And this has provided a unique model to study myeloid cell differentiation. We also were able to, and he was able to extend this to looking upstream at upstream progenitors with a number of additional growth factors to study myelopoiesis in the presence of tumor-derived factors. So this is some work that we're continuing to evaluate and use. So again, as I said, these monocytes derived from this in vitro model with tumor-derived factors, so these GWA or the early passage cell lines, and what you can see is when there's a co-culture between the monocytes that have been co-cultured with the melanoma, there's a suppression of interferon gamma production by stimulated T cells, and this is a contact-dependent mechanism. And then when we look at NK cells, what was found was that these NK cells was prostaglandin E2 that I mentioned earlier, being this central factor secreted by tumors, that by co-incubating the monocytes with prostaglandin E2, that activity suppressed natural killer cells. So work is ongoing to really try to understand more closely, and has been in the lab and continuing, about how to look at how we can inhibit prostaglandin E2, and it's since it's so central. So some recent work, I'm going to skip one here. Some recent work has really addressed this as a combination therapy. Last year, this work has looked more closely at COX-2 inhibition with these checkpoint blockade therapies, and found that actually these actually enhance the ability of the immune system to stimulate decreased tumor size. And this is really bringing up a broader issue, the role of prostaglandin E2 in tumor microenvironments, basically this connection to ongoing inflammation. And it's been long known that inflammation is a risk factor for cancer, and this issue of the inflammatory environment of the tumor microenvironment, by blocking PGE2, we're decreasing the inflammation and then restoring type 1 interferons activity to decrease tumor size. And now this is really connecting with a lot of other pieces, one is the importance of the type 1 interferon pathways and often the reciprocal relationships between inflammation and type 1 interferon pathways, and is connected actually to the ability of dendritic cells to prime T cells. So this is ongoing work. And in summary, some of the work and some clinical trials have gone on and seen some success targeting myelopoiesis. And this is an area of ongoing research in the lab to skew away from MDSCs and to dendritic cell and macrophage differentiation. So in a broad view, chronic viral infections induce some very similar pathways. Myeloid-derived suppressor cells are increased during HIV infection. Dendritic cells are decreased in number, as I've talked about in cancers. And we also see this in sepsis. MDSCs are really central in understanding the mechanism by using this in vitro model, as well as other models as one really ongoing area in the lab, hopefully to address chronic inflammation and the role of the myeloid cell compartment. So I want to talk about one other area of research in the lab, just some preliminary results that are unpublished, about using a drug that's historically been, it's a bisphosphonate drug called Zoledronic Acid, that's been historically used for osteoporosis or advanced stage cancers that have pathology in the bone, osteoclast. It's an osteoclast inhibitor. So I want to tell you a little bit about that in some of our work. So we looked in cancer patients. So these were cancer patients, solid tumors, advanced stage. And what we saw in the blood was a decrease in T regulatory cells in the blood, an increase in the proliferation of conventional T cells, and an inverse relationship between the percent of Tregs and the ongoing proliferation of conventional T cells. When we looked at NK cells in the blood, they had increased production of gamma, of interferon gamma, and then a similar relationship to Tregs. So we treated Tregs overnight with zoledronic acid and saw, looking at the FOXP3 transcription factor denoting Tregs, we saw a decrease in Tregs after zoledronic acid. And so then we decided to use these Tregs that we stimulated overnight in genomic studies to try to dissect what pathways were actually altered in the Tregs by zoledronic acid, or at least converting away from that phenotype. So in summary, we would say that there was a modest effect transcriptionally. We found about 500 genes that were up, 100 or so that were down regulated. So these were up and down. We treated 6 donors with plus or minus ZA in the presence of some other stimulations. And in summary, what we found was, here's a checkerboard plot. I know it might be a little hard to see. The top 2 pathways, there were genes of the calcium channel activity that were altered in all of the donors. We did some calcium mobilization assays and saw that there was less calcium mobilization in the ZA-treated Tregs. And the importance of calcium mobilization in Treg activation is really critical, as INFAT and FOXP3 colocalization was decreased with ZA treatment and is quite dependent on calcium mobilization. So in summary, I wanted to just say that these are all of these areas of these immunosuppressed cells that are found within tumor microenvironments that are being proposed in the field to use in combination with checkpoint blockade immunotherapies that relieve the exhausted, which is a new term for the exhausted T cells in the tumor microenvironment, and then also play a role at targeting some of these different cell types. In summary, our future directions are to really use this in vitro myeloid cell differentiation model. Didn't finish the bit. And to use this further, look at myelopoiesis, as well as continue these ongoing studies. So this is my group at Nova Southeastern University. I didn't mention I'm part of the Center for Collaborative Research, as well as the Cell Therapy Institute. And the lab members are Dr. Julia James, who's here today, and Ron Thomas as well in the lab. And all of the work that I presented here is done in collaboration with Andreas Lundqvist. And I want to send a special thank you to all the Karolinska Institute visiting scientists, some of who are here today, and as well as Dr. Richard Jove, Dr. Aldilduru, who are also here today. Thank you.
Thank you, Shannon. That clearly illustrates the importance of the immunosuppressive tumor microenvironment for all types of cancer immunotherapy. So we're going to, I'm sure, be hearing a lot more about that. Next is Dr. William Decker. He's an assistant professor in the Department of Pathology and Immunology at Baylor College of Medicine in Houston. And the title of his talk is Treatment of Spontaneous Splenic Hemangiosarcomas in Outbred Dogs with Immunologic Stem Cells and Implications. So please welcome Dr. Decker to the podium.
Okay, thank you all for being here today. I'd also like to thank the organizers of the conference, Bernie and Alan, for having me out here. It's always a privilege to be here and share what we're doing with this audience. In the interest of time, let's see here. These are my disclosures. They're important. Look at them. I'm conflicted like most people here, so pay attention to those. I think in the interest of time, we can skip the lecture on the history of cancer immunotherapy. We can also skip the lecture on cancer to stem cell disease and fighting fire with fire, which is the joke I make here every year. I think we can even, because now I don't believe this is really just a regenerative medicine conference, it's really a cell therapy conference, so I no longer have to justify my presence here by trying to convince you that dendritic cells are some kind of stem cell, although they really are. Dendritic cells really are kind of an immunological stem cell in the way that they're able to manifest vast plasticity in response to the environmental stimuli that they receive. So the goal in cancer immunotherapy is to generate a Th1 response. Now, I apologize to the non-immunologists out here in the audience. I'm going to lose you. But by the end of the talk, I promise I'll bring you back. So this is one of my favorite slides. It was a survival analysis done in colon cancer patients. So you can see that patients that developed a high, what we call Th1 response in conjunction with therapy did great. But however, patients that did not develop a high Th1 response in conjunction with therapy did poor. So it's our goal in cancer immunotherapy to generate this Th1 response thing. So what is this Th1 response thing? So basically, Th1 response is cell-mediated immunity, and Th2 response is antibody-mediated immunity. These are broad paradigms. In general, we think that we want cell-mediated immunity, NK cells, CD8 cells, certain types of antibodies in order to get a good anti-cancer response. So how do we get this Th1 thing? I've taken that slide out, so I'll ignore that entirely. So I want to briefly touch upon an observation that I made quite some time ago, something like 12 years ago. Really? Did I leave that slide out? That is horrible. Okay, so in our search for factors that control the Th1 response, there are a lot of different canonical things known to regulate Th1 responses. These are thought to be largely type 1 interferons, patterns that are associated with viruses. However, we made an observation many years ago, and in the interest of time, I can't really go into this observation. However, the observation led to the following hypothesis. So in generating a Th1 response, what you're really doing is addressing a viral response. So you're trying to recognize characteristics that are unique to viruses. And one of the really unique if the antigenic environments on the inside and the outside of the cell were the same, and if such a mechanism existed, would that actually be important? Would that be some kind of key to promulgating Th1 immunity? So again, in the interest of time, I'm going to skip the fancy slide where I show you that stochastically this is actually very feasible. Inside the dendritic cell, proteins that are made inside the cell get chopped up and bound on something called MHC class 1, and proteins that are on the outside of the cell get sucked into the dendritic cell, get chopped up, and presented on something called MHC class 2. So what this slide is meant to show you is that actually class 1 and class 2 come together in close proximity, where if such a comparison could take place, it would be at this step. So to test this hypothesis, and this is all very old work that I'm showing you at this point, and this was published quite some time ago, we did a very simple experiment. I basically ground up cells into two component fractions, lysate, which we threw on top of the dendritic cells to be taken up as class 2 antigens, and mRNA, which we electroporated inside the dendritic cells, after which the mRNAs were translated and then chopped up by the normal proteolytic processes to be presented on MHC class 1. So what I did is I would either take a single cell type and divide it up into lysate and mRNA subfractions, or I took very disparate cell types that had very different antigenic contents and chopped them up into their lysate and mRNA subfractions. And then I mixed and matched them with dendritic cells, and I looked at the outcome with regard to Th1 immunity. So long story short, when the internal and external antigenic environments of dendritic cells are the same, you get what looks like a massive Th1 response in the absence of any other kind of pattern recognition or interferon response. So what we did here is a microarray. We did that back then before mass sequencing was invented. And what we saw was a very unique, specific Th1 profile generated by dendritic cells that were loaded with internal and external antigenic environments that are the same. If you load the dendritic cell in any other conceivable way, you do not get this very specific transcriptional profile. Two incredibly important genes came out of this and we've been working on these two genes now for close to a decade. And the names of these two genes, one of them you've all heard of, or at least you should have heard of if you're cancer immunologists, which I assume most of you are, right? So, and that thing is called CTLA-4. So you will be surprised to hear, however, that CTLA-4 is being expressed by dendritic cells, because most people don't know that. The other interesting gene that came out of this profile was this thing called AMP1. And what we discovered by lots of different experimentation is that when we generated Th1 responses by means of matched internal and external antigenic environments, a lot of AMP1 ended up being secreted from dendritic cells. And at the same time, the constitutive release of CTLA-4 from dendritic cells substantially declined. In fact, it ceases almost completely. So we call this the CTLA-4 AMP1 immunologic access, and you've never heard of it, but you're going to hear about it because it's actually a critical process upstream of IL-12 that drives Th1 immunity. Do I have time to go into this? So sure, I've got time to go into this. So this is actually the sensor complex that we think scans MHC class 1 and class 2 and looks for class 1 and class 2 peptides that are identical. And it just happens to turn out that this AMP1 thing I told you about in the previous slide is a key structural component of this complex. So the hypothesis goes that when this complex detects class 1 and class 2 antigenic matches, there's a conformational change, and this AMP1 thing is released, after which it signals. So I'm just going to show you a little bit of data. This is some new data that the lab has generated recently. So this is what happens in an AMP1 deficient mouse. You can give a mouse cancer. These experiments were done with AMP1 knockout dendritic cells or wild-type dendritic cells. So what you can see here is you can give a mouse a melanoma tumor that has a model OVA antigen in it, and they get quite sick and die. And you can give either a wild-type mouse that has normal T cells or an AMP1 knockout mouse that has deficient T cells, AMP1 knockout dendritic cells, and you can see that neither of these mice can control their tumors very well. However, in the case that you give either an AMP1 knockout mouse or a wild-type mouse dendritic cells that have been loaded with internal and external OVA determinants, you get very good tumor control. So we get very good tumor immunity. in this model system. So the other thing we showed with regard to AIMP1 is what happens with viral infection. So in the absence of AIMP1, low levels of viral infection result in rapid death, whereas in the presence of AIMP1, viral infection is able to be tolerated quite well. This is an influenza model. So the other really interesting thing that we did some genomics. So the people have been doing big data now for decades, and so we went to the TCGA database, and we found 2,400 primary tumor samples for which we could also link to outcomes data in the USC genome browser. And what we discovered is that if you had any AIMP1 expression at all in your primary tumor, i.e., so we have red as high and orange is medium, and green is baseline. So if you had any AIMP1 expression in your primary tumor, by 15 years after diagnosis, there was fully a 71% survival advantage among individuals with cancer who had any kind of AIMP1 expression. We also looked in the Metabrick database. We saw a similar result in breast cancer. We also used the melanoma data set and saw something identical to this. Essentially, if you had some kind of AIMP1 expression early, you got this very good long-term survival, whereas if you didn't, you got much poorer long-term survival, and this was consistent over a very broad variety of cancer types. Okay, the CTLA-4 side of the AIMP1 CTLA-4 immunologic axis is simply as follows. CTLA-4 is constitutively expressed as a safeguard to prevent inappropriate T-cell priming. When CTLA-4 expression is artificially abrogated, as we did here with siRNA, all of a sudden you turn an ineffective vaccine that's not able to control tumor into a very effective one, and you prolong survival substantially over the course of this experiment. So this was the complicated immunological part where I think I lost most of you, but I try to talk about this new immunologic axis that we've discovered and that we've studied. So now we put this into some animal models in conjunction with what we would do in the clinic, which would be, in the case of this pancreatic cancer model, gemcitabine chemotherapy. So what we show here is when we vaccinate the mice in conjunction with some gemcitabine therapy, we get a permanent and durable immune response, whereas if we just give gemcitabine alone, the mice do drop to 0, but eventually all relapse. And of course, if we don't treat them, they all die. So we took these surviving mice that we got after gemcitabine and vaccination, and we re-challenged them many months later, 8 or 9 months later, with secondary tumor bolus in the retroorbit. And what we showed is that the guys that were still alive, who had been vaccinated and got chemotherapy, continued to maintain very robust anti-tumor immunity, whereas some of those guys that had gotten chemotherapy but had relapsed, when you gave them a secondary bolus, they grew giant tumors just as rapidly as naive mice that had never seen anything. So what we showed by virtue of this experiment was that this vaccination approach that we can do in chemotherapy is capable of generating an enduring, long-lasting immunity. We did some immunophenotyping of these mice, and this very, very interesting thing popped out completely by accident. These are CD8 T cells that are also NK1.1 positive, and there was a very strong correlation between these guys and anti-tumor immunity. So then we did a really crazy experiment where we did a massive selection of antigen-specific CD8 positive, NK1.1 positive cells, and we adoptively transferred them into mice that had been not previously exposed and were just given a big bolus of tumor. And as you can imagine from such a massive selection, we had very few cells left over at the end. We were only able to stick 1,500 cells into each mouse. However, those 1,500 cells nonetheless were still sufficient to completely abrogate the tumor that we had injected into the mice, and these mice maintained permanent tumor immunity for the duration of the experiment and beyond. We took this out again many months. So now we know what we're looking for. We're looking for these CD8 NK1.1 cells. There's a human equivalent, CD8 CD161 cells. I wish I had more time to go into the biology of those cells because they're fascinating. So then we did, this is a very recent experiment just done last week, in which, the results were available last week, in which an instructor in my lab vaccinated mice in all kinds of different ways in conjunction with chemotherapy. These guys got nothing. These guys got gemcitabine only. These guys got an mRNA dendritic cell loaded vaccine plus gemcitabine. These guys got a lysate loaded dendritic cell vaccine plus gemcitabine. These guys actually did get our lysate plus mRNA vaccine with gemcitabine, but this is in conjunction with chemotherapy. And what you can see is the combination of the two generates an enormous increase in these CD3, CD8, NK1.1 magic anti-tumor cells. So when we do that experiment, you can see 7 out of 10 of these mice had NK1 circulating CD8, NK1.1 cells over background, and in the other groups, essentially none did. So again, we've got a way to make these really fantastic cells that we know can mediate great anti-tumor immunity. So now we have to really prove it, because we all know that doing things in mice doesn't prove anything. So we turn to dogs. So everyone likes dogs, and dogs are just like little people with fur and tails. So we collaborated in conjunction with local vets to take on this very serious disease called canine splenic hemangiosarcoma. It is a horrible disease. It kills 2% of all dogs. Median survival after diagnosis is only 48 days. So that means half the dogs are dead 48 days after diagnosis. You can see some die right at diagnosis. You essentially have no time to treat these dogs. So anything that you do that can extend life beyond this 48-day time point really should be believed. You can also see that essentially 100% of these animals are dead by a year post-diagnosis. So this is what we decided to do. So this is essentially our clinical protocol. Dogs show up on day 0, bleeding to death through the giant tumor in their spleen. Takes between 5 and 10 days to get the path back with a positive diagnosis of hemangiosarcoma. Meanwhile, what we've done is we've taken their tumors and cut them out of the spleen and cryopreserved them, taken them back to the lab. As soon as we get a positive diagnosis, I harass the owner into signing informed consent and forcing them to mobilize the animal with Neupogen, the same Neupogen we use in people. So these animals get GCSF mobilized to get their peripheral dendritic cell precursors, the granulocytes, the primitive granulocytes into circulation. Day 10, we do a big blood draw, about 250 mils. Between day 10 and 17, we do vaccine production, clocks ticking. And on day 17, the animals get their first vaccine in conjunction with imaging that confirms the presence of disease. For the next 40 days, the animals get type 1 interferon in conjunction with 2 doses of low-dose doxorubicin and 4 additional vaccinations, and we monitor them for up to a year. So this is sort of an anecdote. This was the first animal in our trial. This was the disease we were looking at when we started treating it. This is a 3-centimeter mediastinal mass. You can see over the first 60 days of the treatment, this mass resolved completely. This animal is still alive now at almost a year. This is our Kaplan-Meier, basically. So we have 4 dogs that have either successfully enrolled and completed treatment or are at the latter stages of completing treatment. And as you can see, if we can get the treatment into you, survival is awesome. These animals, even if they have metastatic disease, it resolves and they're doing great. We did lose 1 animal right here, which is good because we don't want the data to look too good or you'd think I'm lying. Already you should kind of think I'm lying because you can see no animals are dying here. That's because I'm not showing you the intent to treat population. You'll be relieved to see that when I show you the intent to treat population, we do lose animals that die in that 20-day period before we can start treatment. So this is what the intent to treat looks like. Still, it looks very good, which convinces us that we may be onto something and that this may be a viable approach in human clinical trials. These are my lab members. They did all of this fantastic work and are to be commended. They're incredible people. There are 1 million people to acknowledge, the people in the lab, other critical people, all of my clinical collaborators who are helping us translate this into pediatric brain tumors, dermal angiosarcoma, and pancreatic cancer. Here are the funders. They've also been incredibly important. Note that there is a lot of foundation funding and some industrial funding. And of course, the veterinary collaborators have to get their own slide for putting the lives of their patients in my hands. This crazy guy that showed up one day and said, hey, let's cure cancer. There are a tremendous number of vets that we're working with. That I learned that word from Donald Trump. That's good, right? Tremendous number of vets that we're working with. We have the surgeons at Gulf Coast, also the oncology department at Gulf Coast, and a number of general practitioners working with us at Sunset. And I hope I didn't go too long, and thank you very much.
Thank you very much, William. Clearly, there are tremendous implications for human treatment. What we heard today in this session was focused on preclinical studies, and which will impact the kinds of clinical trials that we heard about earlier today in the Immuno-Oncology session. So I see we have some questions. Everyone, please go ahead.
Thank you for the sessions. I have one question for Dr. Propa and one question for Dr. Murray. So regarding the GD2 study, do you actually use the anti-GD2 antibody as a positive control in your experiments? So you showed the GFP data, but the antibody treatment arm I missed it. Is it?
It's a chimeric antigen receptor, so
I
it is
get
a
that.
transmembrane protein. We just use
No, I get
yeah.
that it's a CAR
Yeah.
construct, but as a positive control, do you separately, in a separate population, use an anti-GD2 antibody to compare your CAR GD2 T cells?
No.
Okay.
The monoclonal antibody, no. We have just performed studies with the CAR.
That was one of the questions. The other question to Shannon about the so PG2 data is very interesting, I think. Did you ever do an IDO versus PG2 comparison? How the IDO treatment affects these cells as well?
You mean how they affect myeloid, how it affects myeloid cells, or how it affects natural killer cells and T cells?
So IDO treatment of NK cells have been published, but I was curious about the sustaining MDSC activity and suppressive activity on NK cells. How does it affect in comparison to PG2?
Right, I'm not absolutely sure about those. I haven't done those studies, but I'm not absolutely sure about how those compare. I know that by neutralizing prostaglandin E2, you don't get, in the in vitro model, we don't get conversion into MDSCs. And in the more recent studies that have used not the IDO, but the PG2, that is sufficient to decrease the MDSC population significantly on its own.
Okay, any more questions?
I have a question for Shannon. So
Please.
could you maybe tell us a little bit about the literature that exists on common COX-2 inhibitors like just ibuprofen or naproxen for the treatment of cancer? Do those studies exist?
So the studies do exist. There's been a number of trade names of the cyclooxygenase inhibitors, so there's been a history of using those, and they've shown some success. There are high adverse events as far as cardio.
So are these were the COX-2 specific ones, like the celecoxib and Vioxx, or is that what you're talking about?
Yeah. So
right. So I'm sorry. I didn't mean to say COX-2. I'm going to say the global COX inhibitors, like ibuprofen and naproxen. What about those? Not just the COX-2 specific ones.
Okay. I can say that from my understanding is that they've been used to with some success. Some GI tract side effects. I would say. As far as using them right now, the push is to try to use them in combination, not just on their own, although there have been some cancer types that have responded to some improvement with some of those inhibitors.
Okay, great.
Okay, thank you. I have one final question, William, for you.
Sure.
What do you feel are the prospects in terms of treating hemangiosarcomas in humans based on your studies in your animal models?
That is a very good question. So one of the things that we're doing here is conducting, so the neat thing about dogs is, there is no angiosarcoma model in mice, so we're able to actually do some real research here that's been lacking in the field. I have a collaborator at the MD Anderson Cancer Center, Vinod Ravi, who is eager to try this out in dermal angios because that's a disease that has very poor prognosis if you relapse. So one thing we've done here is shown that this is highly feasible, that the logistics aren't too ridiculous, and that we can actually do the whole thing in a reasonable amount of time. So I'd say the prospects are very good, and he and I are currently writing a clinical protocol so that we can hopefully get that through the Anderson bureaucracy and off to the FDA and see if we can get some kind of similar result in people. We'll have to see, though.
Okay. Thank you. Shannon, the final question.
So,
Sure.
Really interesting work, the dendritic cell work, I guess. 2 questions, just your thoughts about cross-presentation pathways.
Yes.
And 2...
My favorite question.
I know, I figured. And then the second was, there have been a lot of studies with the importance of the type 2, I mean, they're called many different names, MDC2s, the ones that are thought to be specialized in cross-presentation.
Yep.
And their role in elimination of melanomas and needing type 1 interferon pathways.
Right. So I don't know if you caught it, but part of this clinical protocol is to supply copious amounts of exogenous interferon. That's critical to the success. So there's a whole process. And if I had more time to explain everything we do, I could talk more about the role of the interferons, basically mimicking what plasmacytoid dendritic cells do. So here's the thing about cross-presentation. So when we started doing this a long time ago, the reviewers all wanted to know about cross-presentation. They'd say, well, no, you throw some class 1 antigens in, you throw some class 2 antigens in, they all end up wherever. It's all the big black box. So we did quite a few experiments, and what we showed really was that cross-presentation is pretty biased. So you can take an antigen and throw it on top of a dendritic cell, and you can get cross-presentation. So it really makes it onto MHC class 1. You really do get some T-cell responses. That really happens. However, endogenous class 1 antigen presentation is much, much more robust. So the things that actually start out in the class 1 compartment are far more likely to end up on class 1 than stuff that comes in. So it's not that I discount cross-presentation completely. It's obvious it's a very important mechanism, but when we throw things in on the outside, and we do see CD8 T-cell responses against it, they're not the same kind of responses. So we just don't see these CD8 NK1.1 cells from things that derive from cross-presentation. So I would say you're still getting a CD8 response. You're getting one level of CD1 immunity, Th1 immunity, but the difference is in durable memory. I think that these durable memory responses have to be produced you need a lot of different signals, but I would argue that this internal and internal antigenic environment signal is pretty critical for durable memory. It's my long-winded answer.
Thank you. We have time for one last final question. Please, Hans Gustav.
Just a very short question. I'm a little bit intrigued by the cellular immunotherapies in dogs. It's really interesting. It's of course both in a way veterinary medicine, but it's also feasibility studies for humans. Do you produce your cells under GMP conditions, or what are the requirements?
So we do have a GMP protocol for our human clinical trial. So my 2 people in my lab are trained in GMP. They go into the GMP facility, use GMP techniques, FDA approved clinical protocol when we've done all our preclinical stuff. We actually have not been required to use the GMP facility to do in this circumstance. The IRB at Baylor didn't require it. The internal IRBs at the vet places didn't require it. However, I will say the however is that my people have been trained in GMP, and everything else we do with those dogs is at the level of GMP. We just have been able to avoid officially having to use GMP. Well, we do really use GMP grade reagents, to be honest with you, because these are people's and pets. We're taking their lives in our hands. So we haven't been required, but the only way in which we're not doing GMP is basically we're not actually walking into the facility. We're keeping it in a specialized tissue culture room that's more GLP than GMP.
Great. So thank you. I'd like to thank the speakers for very stimulating presentations and discussions, and I'd like to thank the audience, everyone, for attending. And I just want to remind you there's still some more activities, and as you head home today, travel safe, and thank you all very much.